Hello and welcome to our channel Bioenigma Uncovered. In our previous video, we assigned taxonomy to the representative sequences or the ASVs using the Silva v3 v4 classifier. In today's video, we are going to generate taxa-bar plots of those ASVs to visualize the microbial composition of our samples. Before we proceed, it's a humble request to please subscribe to our channel, as more than 80% of our viewers, they watch our content but do not subscribe. Let's start today's video. To create taxa-bar plots in QIIME2, we need three files. The first is table.qza, which will provide the abundance data. Second is taxonomy.qza file, which will provide the taxonomic classification or the classified representative sequences. And the third is sample metadata in the form of metadata.tsv. Now, as you can see on your screen, we are going to type the command QIIME space taxa space bar plots. So let's do it. First, you need to basically locate all your files which are to be present in your appropriate drive. Fine. So you need to locate that and just press ls. In the next step, we are going to type the command to generate the taxa-bar plots. As you can on your screen, QIIME taxa space bar plot space press space backslash enter, then press the type dash dash I means input. You need to first input your table, so you need to first check what is the label of your table.qza file. So you can see it is right here, that is table.qza. So just select that and paste it here and space backslash enter. In the next line, type dash dash I dash taxonomy space taxonomy dot qza. Again, it is very critical to have your file name exactly as it is in your file or folder. For convenience, it is always suggested that you must copy paste the file name and just put it over here. Okay, fine. In the next step, you need to input your metadata file dash dash m dash metadata dash file and then paste your metadata file. Remember, your metadata is a tsv file. Just put it over here, space backslash enter dash o dash o dash visualization. Remember, it is highly recommended to type the spellings very critically, and it is always advisable that you must speak while you are typing. This will minimize your typing errors. And then just type the taxa output file taxa-bar-plots.qzv and then just press enter. Fine. So one thing you must understand here is that this table.qza file, this will provide you the information about your abundance data of your various representative sequences. In the next case, that is taxonomy.qza, it will provide information about the taxonomic classification or the hierarchical information about the various taxa, and the metadata will provide information about your the sample records. So as you can see right now on your screen, your taxa-bar-plots.qzv file is now generated. In the next step, we are going to analyze taxa-bar-plots.qzv file into the view.qiime2.org website. Now let's move to that web page. Open your browser web page and type view.qiime2, write the spelling name very correctly, QIIME2.org, press enter, then press on drag and drop or click here, locate your file, press open. Once it is open, you will see that there are a lot of vertical bars present. Each bar represents one sample. So we can just increase the width of the bar for better demonstration. So as you can see here, there are 11 samples for this particular example being under consideration. The height of the bar represents the relative abundance. So it means, and this is present in the form of percentages. So it means that it is not going to give you any count of the reads, but it will this bar or this graph will provide you information about the relative abundance of each or individual taxa, ranging from division to genus or species level. So right now this green bar is representing the division bacteria, as I am showing here on your screen in this highlighted text. So you can increase the bar width. In addition, you can change the colors accent also, like I can just darken it here for demonstration purpose. And besides, I can also sort the samples based upon the metadata provided. So like the sample name, version, creation date, release, there are a lot of data. So this thing, like if I change the geolocation underscore name underscore country, it is showing that study was carried out in China. So right now we will move to the very default position, that is samples have been sorted on the basis of the taxa, that is division. So this is the very first level of taxonomical hierarchy which is being shown here. In addition, if we move down to level 2, this graph is going to provide you information about the phylum taxa as well. So if I click on this just green button here, it is showing that the relative proportion of phylum, this phylum Firmicutes. Okay, so just click on, and if I just show it on this purple box here, it is providing us information about the phylum Bacteriodota. What do you see here? It is that this sample one, which is SRR 21601759, it is having the highest proportion of the phylum Bacteriodota, whereas last sample, that is SRR 21601753, it is having the least proportion of the phylum Bacteriodota. So if you just highlight only each individual box, it is going to provide you information about the relative abundance of each taxa. You can see here that phylum Actinobacteriota, it is not represented in two of the samples, that is 1759 and 1752. Moving down to the taxonomic hierarchy, in addition to division and taxonomy, you can also like check the class which is presented here. So again, if I click on, so here in this orange color, it represents the division bacteria, phylum Firmicutes and class Bacilli. So you can see here that class Bacilli is present, or it is represented in all of the samples under consideration. Now, if I click on this, you can see this actinobacteria is not present, or it is present only in one sample, that is 1761. By selecting all these levels, taxonomic levels, you can come to know, you can have the information about the relative abundance for all the taxa, ranging from division to genus levels. The division level taxonomy provides information about the broad taxonomic hierarchy across all the samples. As we move down across the hierarchy, that is from division to phylum to class to genus to species level, you will see that the abundance of each individual organism, or rather each individual microbial signature, can vary substantially across all the samples, as you can see right here on your screen. So as we move down to the genus or the species level, we can have information about are there any newly discovered microbial signatures, or if there are certain genus or species which are overrepresented or underrepresented across different samples. One another important point that you must remember is that these taxa-bar-plots arise from ASVs which were generated during the DA-DA2 denoising step. So if the fraction of those ASVs generated during that step is below the threshold value of 30 to 50%, then your taxa bar plot will provide you incorrect information. More important points that you must also consider while generating these taxa bar plots is that if your metadata file has errors, then this taxa bar plot will fail. If your taxonomy.qzv file has some unassigned taxonomic hierarchy or unassigned taxa, then the same will translate into some unwarranted errors as well. So you must take care of all these things before reaching to these steps. So the success for generation of these taxa bar plots will in turn depend upon your DA2 denoising and the filtration step, which is the most critical step when executing the QIIME2, when executing or analyzing your data through QIIME2 platform. So in the next step, we are going to analyze the diversity matrix in the form of alpha and beta diversity from these taxa bar plots. If you do end up like our content, then please don't forget to hit the bell icon and also please share it with your friends and your peers. I'll see you in our next video. Till then, take care. Have a nice day.